So your DNA helicase, which is this DNA helicase, this basically opens up the DNA strand. Okay, so we have, let's change color. It's like this. And then you have your nitrogenous bases, G, C, C, whatever, T, all that fun stuff, okay? And then over here, there's gonna be a little section right here, which is called the promoter. Remember that one for later. Promoter is really important for later. So, what happens is there is also one more thing. Let's just get an orange. Yes. Okay, there is something here. Alright, and this we're gonna call a transcription factor. We talked about that before. Factor. So far we have the DNA helicase opening it up, the transcription factor that is starting the whole transcription in the first place, and then we have our promoter, which is a section before the gene that signals for the transcription factors. And then we have one more thing. I'm gonna do it in pink. We have it right here. And this one is gonna be the RNA polymers. Polymerase, which is that enzyme that we talked about that forms the covalent bonds for the RNA strand as it goes. So this promoter is gonna go over here and it's gonna start forming bonds. So this right here, it's gonna be a, let's see, we have TA CG, okay? So this RNA polymer is going to come here and it's going to start collecting. It's going to make an A, it's going to make a U, it's going to make a G, and then it's going to make a C. It's just going to bond them all the way up here. So let's add another T and A and then a G. So it's going to keep bonding them. And then you're going to end up with like a strand of RNA over here. So this, what happened? Oh, okay. So the strand is gonna finish over here. Okay, the RNA polymer is gonna stop right here. It's gonna stop at something called the terminator. So the terminator is gonna be right here at the end. Terminate. Okay, that does not look. Terminator. Okay. And after it stops at the terminator, you're just gonna have a RNA script of whatever DNA you had before, which is this pink one. So, back to this. We have initiation, where the DNA helicase breaks the hydrogen bonds between the nitrogen bases. So these hydrogen bonds right here, they're broken, they're gone, and then we build these new ones right here. And then the transcription factor, which is this right here, and the RNA polymers, they bind to this promoter right here. And this identifies the start of a gene on one template strand. So this is called a template strand. So, transcription factor, RNA, bind to the promoter, identifies the start. And then we have our second part, which is elongation. So in elongation, the RNA polymers, which is this one right here, the pink dot, the RNA polymers is going to position complementary RNA nucleotides along the DNA template. So this is the DNA template. This is the RNA nucleotides. It's going to place them on here, and it's going to join them with covalent bonds. So A to T, U to A, G to C, and this is going to create a RNA transcript. So let's put this right here. Transcript. Perfect. Then the last stage, oh here, let's write it here. So the RNA nucleotides, they're gonna join the DNA template with hydrogen bonds. So that takes us to our last step, which is 3, and that is termination. So in termination, this is where the polymers, right here, it reaches the terminator on the DNA, which indicates the end of a gene. So the RNA polymers and the completed RNA transcript, they're going to detach from the template DNA, and then transcription is complete. So we're going to end up with something like this. And that's going to be our RNA transcript, which is pretty much a copy of this one up here. And that is termination. So that is the process of transcription. There is one last process that is important. Remember how up here, do you remember how up here we said that after it's an RNA script, it cannot leave the nucleus until it is an mRNA? So we're going to talk about how it becomes an mRNA from an RNA. So before it leaves the nucleus, it has to become a mRNA. And why is this important? It's because enzymes in the cytoplasm, they can attack and eat this RNA if it doesn't have like its proper identification. So think about RNA processing as if we are putting kind of like a label on it, telling the cytoplasm, the enzymes in the cytoplasm, to leave this one alone. Tell them that, this one is with us. So each DNA has something called an intron, which is kind of like its junk. And if that stays in the DNA, it will result in incorrect amino acid sequence. So it's going to make the wrong protein. And there's another part in the DNA called exons, which is important for correct protein structure. So what we need to do, basically, is we need to label the RNA transcript, we need to remove the junk, aka the introns, and we need to keep the exons that will help us get the right protein. So those are basically the steps of RNA processing. So RNA processing. And let's bring knockouts down here. Okay, this is the last step of transcription. So there are 4 steps to RNA processing. Step number 1 is called adding the cap and tail. So cap and tail is added. So basically, let's try it out actually. So we have, let's do this. So we have, okay, and then we have this part, and this part, and then this part, and then this part, and then this part. Okay, So the red is the introns. The blue is the exons. No. The blue is the cap and tail. And then the orange is the exons. So in the first step, we add these. We add this tails, the cap and tail. And then in the second step, we remove introns. And these introns, they get recycled to make DNA and RNA all again. So these basically exit. We get rid of the introns. So it leaves us with something like this. So we have our blue and then our blue. Okay? And then our third step is to splice the exons back together. Think of it as in like we're stitching them back together. So we splice the exons together. And then when the exons are finally spliced together, when the exons are, one second. Gotta let my cat in. So when the exons are finally spliced together, it's gonna look something like this. Oh my god. Bear with me, guys. And then the blue. So it's gonna look like this. This right here is completed mRNA. This can finally leave the nucleus. So that is our last step. Step 4, mRNA leaves nucleus. And it leaves through the nuclear pores, and it exits to the cytoplasm, and then this mRNA will join a ribosome, either in the cytoplasm or in the rough ER, and it's going to begin the process of translation. So that is the process of transcription. Pretty simple, pretty easy. What do you think, Nagops? Did you get it? Do you understand? I think she understands. And yeah, that is it for today. We will see you in the next video.